Hello, everyone, and welcome to today's webinar sponsored by the HIBAR Research Alliance, also known as the HRA, and produced in collaboration with the National Academy of Inventors. I'm Scott Slovic from the University of Idaho, and I'll be the moderator for today's one-hour event. This is the 12th in our series of webinars for researchers, research administrators, and others who are interested in what we call highly integrative, basic, and responsive research. Our previous webinars are now available for viewing on the HRA website, which can be found at hibar-research.org. A recording of today's webinar will also be posted on the HRA website. Before our featured presentation by Drs. Peter Cullis and James Taylor, I'd like to explain the structure of today's meeting. We'll begin with a few words of welcome from Jamie Renee, the Executive Director of the National Academy of Inventors, who's helped with the planning of today's event. We hope to work with NAI in developing future webinars as well. Next, there will be a brief overview of the HIBAR research concept by Dr. Lauren Whitehead. Peter and James will speak about the HIBAR aspects of their research on lipid nanoparticles in relation to COVID-19 vaccines and genomic medicines for between 20 and 25 minutes. And then we'll have approximately 20 minutes for Q&A, followed by an opportunity for our presenters to offer a few final takeaways. Now let's get started. Jamie, the microphone is yours.
Thank you. Well, I'm delighted to be here with you guys today, and we've got an exciting topic that we'll be going over. If you're unfamiliar with the Academy of Inventors, I encourage you to go to our website at academyofinventors.org. The National Academy of Inventors was founded in 2010, and we're an academic organization focused on enhancing the visibility of academic inventors nationwide. We're comprised of 5,000 individual inventors spanning over 250 institutions. We've got curriculum similar to the IP curriculum that you can get free of charge on our website. We also put webinar series out like the one you're attending today, and we have a game mentoring platform. But there's much, much more to us. And so if you're an academic inventor and you're curious about the National Academy of Inventors, I encourage you to go to our website. We'd love to hear from you. Thank you, and back to you, Scott.
Thank you, Jamie. Dr. Lauren Whitehead, the director of the HIBAR Research Alliance, is a professor in the Department of Physics and Astronomy at the University of British Columbia. He'll briefly introduce the concept of HIBAR research and the HRA organization.
Well, hello, everyone. It's so nice to have you here and have just a few minutes to say a little bit about HIBAR research and also the HIBAR Research Alliance. So I'm going to answer first the question, what is HIBAR research? And the key thing about HIBAR research, which you already heard stands for highly integrative, basic, and responsive, is that research projects of this type comprise two key dualities, and I'll explain what I mean by that. The first duality is that the project is led both by non-academics and academics. And the second one is that its purposes include both societal benefits and new knowledge. So to put that in a sentence, in any HIBAR research project, leadership comes both from outside the academic system and from within it, and the purpose is both to yield significant societal benefits and new knowledge. And of course, we aren't talking about these dual dualities because we think they're cool. We are talking about them because for a very long time, it's been observed that projects of this type are highly generative. So let's just talk about that a little bit more. Let's say there's a research topic of interest, somewhere in the world. What we are saying is that if we apply these four lenses of academic leadership and societal leadership and academic purpose and societal purpose, great things happen. And that's what HIBAR research is about. So we use the term highly integrative just to stress the large integration that you see here between the academic world and society. And also the words basic and responsive indicate that the research is not just at one end of the academic spectrum, but spans the spectrum in a highly integrated way as well. So that's the definition of this research. And that leads to the question, why is there a HIBAR Research Alliance? What is its mission? So the Alliance itself is a network of volunteers, some of whom you'll be meeting today. But they're all across not just the United States, but also Canada and actually elsewhere in the world as well. And they're attracted to the organization because it's well organized to do something. And by the way, the organization comes from the member universities that you'll find on our website, and also other organizations that are kindly contributing expertise as well. So the goal of the HRA is to increase the prevalence of university HIBAR research from what we think today is about 1 project in 20. Very good work underway today, has been for quite a long time. But as you've seen here on the screen, to increase it to about 1 in 5 by approximately the year 2030. While also improving all types of research. This will help universities respond more effectively to societal needs, while also increasing the excellence of their fundamental research. So clearly, this webinar series is one of the things that we can do to help communicate about HIBAR research, providing excellent and exciting examples. And it's just so exciting to introduce the research that we'll be discussing today. I'm not going to introduce the individuals. I'll hand back to Scott, but I will basically summarize the connection between what I have just said and the work that you'll be hearing about today. So there were 4 key lenses that I mentioned earlier, and for each of them, I'm just going to talk about today's research. So the work is discovery oriented. In this case, there was a goal to develop deeper understanding of the nanoscale physics and chemistry of encapsulation of substances within hollow nanoparticles. In addition, there was tremendous and is intended to be far more in the future, significant public impact. So likely everyone in this meeting, and billions worldwide, have benefited from the COVID-19 vaccines that were enabled by the work we'll be hearing about today. And that is just the beginning. We're at the beginning of an amazing era of new medicine. Regarding societal co-leadership, well, we actually have presentations from people in both society and academia here today for the presentation. On the societal side, the work took place in partnership with leading members of the biotech business community, who played key roles in helping to guide this success. And in the academic world, within the academic community, key leaders accelerated discovery of the underlying physics and chemistry of the key novel nanoscale phenomena that make all of this work. So I'm just incredibly excited to see this wonderful connection and to have representatives of both of those worlds here to discuss these dualities in the work. And on that note, thanks for your attention. And Scott, I will hand back to you.
Thanks very much, Lauren. And now I'm pleased to introduce Peter Collis and James Taylor. Peter is a professor of biochemistry and molecular biology at the University of British Columbia, and he was recently named a 2022 Canada Gairdner International Award laureate for his work to develop the lipid nanoparticle delivery technology that has led, among other things, to mRNA vaccines for COVID-19. And James Taylor is the co-founder and CEO of Precision Nanosystems, a Vancouver-based commercial stage biotechnology company that brings together nanotechnology, genomics, and precision medicine. Peter and James, the microphone is yours.
So, yeah, I'm going to talk a bit about HIBAR research and lipid nanoparticles and kind of cover a bit of my career. And James is going to follow up with some of the possibilities that are being enabled by this technology, which are quite extensive. HIBAR research and lipid nanoparticles. I have a number of conflicts of interest with Precision, Acuitas, and Nanovation Therapeutics, all of which I co-founded. Now, as Lauren summarized, the HIBAR research is highly integrative, so there's a diversity of leadership, also basic and responsive, working towards trying to solve an important societal problem. And I guess according to these criteria, I've been doing HIBAR research for quite a long time. You can call me a scientific entrepreneur. In a way, I spent most of my career working half-time in academia and half-time in companies. This goes right back to about 1978 when I joined the biochemistry department here at UBC, and then spin-offs of the Canadian Liposome Company, Inex Pharmaceuticals, and then 3 not-for-profits, the Center for Drug Research and Development, Personalized Medicine, and Nanomedicine, all the while performing the usual act. This has caused a certain level of schizophrenia in my life, and we can talk more about that later. So I've done about 10 or 11 startups and 3 not-for-profits. So these companies now employ more than 500 people in Vancouver. So these are starting with Lipex Biomembranes and going through to INEX with Precision Nanosystems, which you'll hear a lot more about, and Acuitas Therapeutics, which is the company that actually did the heavy lifting on the lipid nanoparticle that's used in the COVID-19 vaccines, particularly the Pfizer-BioNTech one. My most recent company is Nanovation Therapeutics. So this has led to 5 approved drugs, 3 of which are small molecule delivery systems for small molecules. 2 of these have very direct utility in the cancer chemotherapy. And then 2 drugs have been approved that involve nucleic acid-based therapeutics. And one of those has been mentioned is Comirnaty, the Pfizer-BioNTech COVID-19 vaccine. Just a reminder that the LNP stands for lipid nanoparticle. In addition to that, there's been a couple of instruments that we've developed for manufacturing, starting with an extrusion device that we invented in the mid-1980s. There's actually more like 10,000 of these have been sold worldwide for making liposomal systems, and then microfluidic mixing, what James will talk about a bit more. That's led to the nano assembler and is being commercialized Precision Nanosystems. So this is the extruder. There are now $1 million versions of the extruder. We started off with a $2,000 or $3,000 one we sold to academic scientists. Similarly, the nano assembler now has a range from bench level right through to scale up for potential clinical use. So how did all this start? The highly integrative HYBAR research engages society with diverse leadership. And so this goes back, as I said, to the mid-80s. We established a collaboration between my UBC laboratory and a company called the Liposome Company based in Princeton in New Jersey. And the Liposome Company was formed to deliver cancer drugs using liposomal systems. The problem was that A, they couldn't make liposomes, and B, they couldn't load them. So it turned out that we supplied both of those needs with the extrusion process that you saw on the previous slide, as well as a method to load cancer drugs into liposomes, which I'll describe in a little bit more detail. Our solution here was what you might call pH loading. And we've been in a very basic research. As I say, Ed needed a way to load cancer drugs. We've been conducting very basic research, looking at the effects of pH gradients, ion gradients across lipid bilayers, the influence of these ion gradients on membrane transport. And to measure pH gradients, we used what's termed a weak base called methylamine. So this the radio-labeled version of this, a methylamine as a weak base is able to exist in a protonated or positively charged form or a neutral form. And the neutral form is highly membrane permeable. It can move across lipid bilayers very, very quickly. Now, if you have pH gradients, say of pH 4, about the acidity of a lemon on the inside and pH 7 outside, then the neutral form can move across very readily. But of course, it's going to get protonated on the inside and the higher acidity of the interior. And you can do the maths on this. It's pretty simple. And you'll find at equilibrium, the concentration of methylamine on the inside over that on the outside will mirror the proton gradient. So in other words, if you have a 3-unit pH gradient, you'll have 1,000 times higher concentration of the methylamine on the inside and on the outside. So this was pretty profound. It was very useful, of course, for measuring pH gradients. And then one day we realized that many common cancer drugs are weak bases. So this is 4 examples, and there's many more. So we wondered, well, gee, can we load these lipid nanoparticles with cancer drugs just using pH gradients? And what we found was we could load them into such high concentrations on the inside of these vesicles that we would actually get precipitates of the cancer drug. This is an example for doxorubicin, which is a commonly used cancer drug. So we get these little nanocrystals inside our lipid nanoparticles. And this turned out to be the basis of a drug delivery technology that has enabled many, about 10 drugs now that have been approved for treating cancer in various forms. 3 of these we developed that use this basic technology. So it's But you can see that the way we got to being able to load these lipid-based systems, these lipid nanoparticles with the drug, was really the result of some very basic research, which gave us some tools which we wouldn't have otherwise had. In other words, the pH gradient and the recognition that perhaps these drugs, which are weak bases, could be loaded in a similar way as the methylamine. So this is a pretty so basic research is certainly there, and certainly responding to the societal problem of disease in the form of cancer. Now, in the mid-90s, this was again a interaction with another CEO who said, well, I can't raise money delivering cancer drugs in liposomes. He needed something with more sex appeal, as he put it, needed to be doing gene therapy. This entailed, or started us off on really what was a whole new path. It was efficient encapsulation of the of nucleic acid polymers, which are, of course, highly negatively charged, really requires positively charged lipids or cationic lipids. However, there are no positively charged lipids in biological membranes. They're really toxic molecules. And so we had to find a way around this. Again, this is where basic research comes in and a good dose of serendipity. But we synthesized a lipid we call DODAP, which was an ionizable cationic lipid. In other words, it could exist in a positively charged form or a neutral form to investigate lipid asymmetry. This is a property of biological membranes. The lipid composition on one side is different than the other. And so we were using that, we could flip this ionizable lipid across a lipid bilayer using pH gradients. And so we could investigate some of the consequences of lipid asymmetry. Well, when we had this problem of loading nucleic acid polymers, we asked the question whether we could load the nucleic acid, the pH where the ionizable lipid is positively charged, and is it retained when we get to neutral pH? Well, then we had to ask the question, what process do we use to get these nucleic acid-based drugs into lipid nanoparticles? So we needed a mixing technique. You can dissolve the lipids in ethanol, and you can dissolve the nucleic acid in an aqueous buffer. But how do we get it together? So Carl Hansen, this is another, I mean, serendipity does play a large role, as they indicated. He said use microfluidic mixing. I thought, well, we're going to get microliters out, right? So this isn't going to be very good. It turned out that the technique worked brilliantly. And so it's a very good way of formulating this is the lipid in ethanol, the oligonucleotide in water. We're mixing very rapidly, increasing the polarity. The first thing to fall out of solution is a hydrophobic complex. You do this fast enough and you can trap these systems in what we term limit-sized particles, which are the smallest particles compatible with the molecular makeup. It turned out that this was a great way of making lipid nanoparticle systems containing RNA or DNA in general, and it was a scalable or reproducible process, as James will emphasize. This resulted, I met James about 12 or so years ago, and he said, I want to be a CEO of a company. I thought, okay, we got some interesting technology here with this microfluidic mixing approach. And so James said he and his compatriot Ian Ramsey said, okay, this sounds like an interesting challenge. And they've really taken this all the way, as James will explain. So the further improvement of the ionizable lipids has led to the COVID-19 vaccines, really was kind of further improvement of the lipid nanoparticle technology itself. So it's been a pretty interesting story that with very basic research, combined with starting companies and interacting with individuals who are concerned about actually making a product, has been really quite productive. So this is the message. Basic research leads to new tools for such as pH gradients and ionizable lipids for loading various cargoes. And the applications were driven really by societal needs, such as improved chemotherapies and the need to enable gene therapies. So it's been quite a fascinating journey that we've been on, but certainly it wouldn't have happened if we hadn't been really conducting this research that had a basic element and a very applied element. So I'll stop there and let James go on with the second part of this presentation.
Excellent. Thanks, Peter. That was wonderful. And so continuing on, and I would just highlight here, I'll talk about Precision Nanosystems and what we do. But, part of the, high bar in the meeting here is we spent the first 5 years of our life really in Peter's lab. And I would say that Peter was, as he mentioned, very industrial in his abilities and culture and so on. And we took that up. And so for a long time, really under Peter's guidance, and then continue to build and continue to grow in our own careers and the company, and have gained quite a bit of success here. And a lot of the success was driven around having conviction for something. In this case, it would be genomic medicines that really wasn't mainstream. There was no COVID-19 mRNA vaccine that was highlighting the utility of these technologies. And so it was a period of really pushing technology and helping to grow the field of genomic medicine. So our company here, we now have over 100,000 square feet of facilities that we're building out in Vancouver. A very fantastic team, one of the world leading teams in nanoparticle development, developing these technologies based on the inventions that came out of our collaborations and work in Peter's lab. And the mission of our company is to accelerate the creation of transformative medicines that significantly impacts human well-being. We decided early on not to be a drug company, but to work with drug companies to help them to make the therapeutics and vaccines that will define the future of medicine. And we see this as genomic medicine. So this is the delivery of RNA or DNA into cells to really affect medicine at its molecular, or affect disease at its molecular root cause. And there's a big toolbox now of genomic medicine modalities. I think the mRNA vaccine approaches are the most commonly known now in society, but there's a wide variety of approaches that are very useful to really targeting any gene in any way, from silencing proteins that aren't causing problems to expressing ones that are missing or not working, to even now going in and editing problems directly in patients' genomes. And these technologies are enabled by the basis of the research done around lipid nanoparticle technologies, with Peter having a huge impact and really a leader in this field. And so these are, as Peter described, they're about 70 nanometer in diameter, comprising hundreds of thousands of molecules that self-assemble to create the final drug product. And they comprise both the active pharmaceutical ingredient, the RNA component, as well as the delivery component, which would be the nanoparticle. And both of those two components are critical for these drugs to work, and they're very useful. So to enable drug companies in this field, what we've done is to develop, starting with the manufacturing technology, the microfluidic-based manufacturing technology, have since really built out a full stack of technologies that allow a drug developer to really push forward on these medicines very quickly. That includes the manufacturing technology, which we'll talk about in more detail here today, as well as lipid nanoparticle delivery technologies, and then also now RNA technologies to enable these medicines. And then also these drugs are very sophisticated, and we've put together a world-leading team in nanoparticle drug development to help our users to develop these drugs. And so we work with hundreds of companies now, leading pharmaceutical companies all over the world, from the largest of pharma to the smallest of startups. And in areas of infectious disease vaccines, COVID and otherwise, rare diseases, and creating treatments for what is a large class of disease that's stratified very specifically across patients, to cancer, and also just generally into personalized medicine. And we think in the future, medicines will be tailored specifically to the molecular basis of an individual's disease, and frankly, will be manufactured near the bedside of that patient. So very exciting time in medicine. And as Peter highlighted, I won't go through this in too much detail, but the application of microfluidics and this base technology in how to make these drugs has had a huge impact. And so the self-assembly of these hundreds of thousands of molecules into these superstructures that come together non-covalently, controlling that is a huge challenge, and being able to make a reproducible drug is a huge challenge. And so the invention and the development of a microfluidic-based approach, applying to the lipid nanoparticle field, but also polymer nanoparticles, liposomes, as Peter spoke to, is really important. And our contribution at Precision and under our company is taking this technology and really industrializing it, being able to make drugs at relevant levels. We do this through, think of microfluidics being small scales, but using our technology, and particularly microfluidic technologies we developed in the last 5 years or so, allow us to scale from small scale to large scale seamlessly. So we can scale up a single reactor to be making, getting close to liters per minute type volumes, to also being able to scale out where we can parallelize processes to make massive scales. So our technologies will enable the manufacture of microliters to work in cells and petri dishes, to milliliters to work in small animal models, and then scaling up for drug production for GMP manufacturing, using these very fundamental technologies, having a deep understanding of physics, how these nanoparticles come together and to create these important drugs. And I'll just move through this pretty quickly, but the technology really creates a reproducible drug product. That's a very challenging thing to do. There's a lot of steps to make these drugs, and we support our users very deeply in that to optimize these. Being able to measure and the analytics of these drugs is very important and really speaks to the sophistication with them. But lastly, I'll leave you with a societal impact. So Precision Nanosystems in where we are in Vancouver in Canada has really been an advocate for supporting the local industry here, growing here within the biotechnology community, but also to support a need of a country like Canada to have sovereign vaccine supply, and supported by the Canadian government to set up biomanufacturing here in Vancouver to support local manufacture of vaccines for future pandemic preparedness. It's a very important technology. Thank you for having us join the meeting, and we'll leave it with that.
Thank you very much, Peter and James, for that crash course in liquid nanoparticles and genetic medicine. Speaking from the perspective of an English professor, I actually found it very manageable to follow what you were saying, and really quite fascinating. I wonder if we could transition as we move to the Q&A part of our webinar from thinking about biological membranes to considering what I might call cultural membranes, the relationship between different segments of society. And so I'd like to begin the Q&A as I wait to see what other questions might be coming in from our attendees, by asking you a little bit about cross-sectoral partnerships and maybe dig a little bit deeper into the relationship between the two of you and your colleagues. So I wonder if you could think about any advice that you would give to a faculty member who's initiating a HYBAR project in terms of making sure that their partners are adding value intellectually and are truly engaged in the basic decision-making processes of a project. So for instance, what steps might you take to make sure that all of the research partners are participating in and fully engaged in the decision-making of a project?
I'll have first go at that, and then James can chime in. But it's a difficult problem. It's certainly set me along a path of doing startup companies as opposed to licensing things out. My experience with licensing is licensing, say you have something in your lab that looks like it's working very well, and licensing that to another company, a biotech or a pharma, is that you really need a champion in that environment to push the project through. And in a couple of instances, I've had exactly the people that are real champions, but then they either leave the company or they get shunted into a different position. And as a result, the licensed technology never gets developed. So whereas if you do a startup company, as a scientist, you can be intimately involved in that. But also the companies are little self-survival mechanism to push all the levers to try and survive one way or another. And so that's really been much more successful from my point of view for really pushing enabling a project to really make it in the commercial world. I don't know, James, what your thoughts are there.
Yeah, no, I would agree, and where I can contribute my perspective of being an entrepreneur, it really is sharpens the pencil, or it really focuses the mind when you can only eat what you kill, right? So you have to be successful, or else you're not around. So you have acute focus on doing something that you plan to do, but also something that is useful for society, because you need to capture value from that utility. And so I think it's a really good setup to be academic and have startup companies. And we've seen this from many colleagues now, And it's a really strong thing for these deep sciences across universities to have that deep tie to the entrepreneurial community.
We have a follow-up question from one of our attendees that suggests that the kinds of input from the societal partner might not only focus on utility, but actually on the deeper research questions, the more fundamental questions. Our attendee asks, how do you filter the new unanswerable questions coming back from the applied side back into the basic research program? Have you found that you've been in situations where this might have happened in your own collaboration?
Peter, maybe I'll start this one and then you can go. But I would say in the life sciences where we sit now, I'm a physicist, Peter's a physicist, we're both in the life sciences though, and it's an industry of deep technology. So it's the challenges being faced come from science. It's a very science-oriented field. So I think industry is taking on pretty fundamental things. And if you look at new companies, therapeutics being started, they typically are having a pretty out there type new approach to problems. And so I think there's a science first approach in the biotechnology industry to begin with. But maybe Peter, how does that feedback into the academic setting?
Yeah, well, I'm just dealing first of all with the pushes that come from the commercial world. Sometimes you have to do you have to say, okay, let's do a reality check here. You may want something that's going to be an IV a system that's going to affect brain function, but it's going to be a little while before we can achieve that. Whereas other tissues are it's much more straightforward. And so sometimes you have to be okay. You can go back to the lab and say, okay, how can we get to the brain? But we know from experience that's going to be a tough one. I mean, there's other examples as well. The art of the possible is always something that that's again where from a scientific point of view, you'd better be cognizant of you're trying to develop a product, and if you set yourself an impossible goal, well, you're going to fail. And so there's a practical element that comes in. The basic science part from an academic point of view, yeah, this is where it gets to be fun because you can obviously go back to the lab and see what you can do in mice, and and you have a few ideas, but you certainly don't promise that as a product to a commercial enterprise. At least if you do, you're liable. It's liable to be. You're not going to be very popular in a few years' time. Let's put it that way.
I wonder if we could talk a little bit about issue of timeframes. Often there's kind of a disconnect between the timeframes of basic research and the timeframe of industry. And I wonder, you've described this multi-decade journey that has led to the rapid success, the overnight success of COVID-19 mRNA vaccines. What advice would you give to others facing a similar long journey to yield substantial societal results, and how to maintain enthusiasm for everyone involved?
First of all, you never know how long a journey is going to be when you start off on it, right? So okay, we started off 40 years ago. The main thing is, are you getting somewhere? And also, have you got a team together? I mean, one of the wonderful things about starting companies is the level of teamwork that you have to get into. I mean, in an academic environment, things are more individually focused. But in a company, particularly a startup company, you really bring people together, and everybody has to do everything. So it's a really enjoyable environment. That for me has always been key, is establishing that really fun environment, and where you're going up against a particular, a difficult problem, and then it's surprising what happens. But it's very difficult, what's the Yogi Berra statement? Predictions are difficult, particularly about the future. You start off with something thinking maybe you can get there. If you have a good team around you, you may not get to the particular objective you had originally, but you'll find there'll be a way through this where you develop a difference, something different. It becomes a really fun journey. I mean, James, you probably can extrapolate on that even more.
Yeah, for sure. I think what I would add though is maybe a little bit of a different angle to it as well. In that with the rise of genomic medicine, this really is the revolution, taking medicine from an analog field to a digital field. And so certain times in history, there's these tipping points that happen. And I think there's a compression now of timescales in when all the development's going to happen. It's going to be an ultra productive time in the decades to come around medicine because of this. And so I think it's a great place to be for researchers at this intersection and to get involved. And we're involved with so many researchers now that are really moving extremely fast on highly disruptive, innovative new approaches to medicine.
Second that. I mean, this is almost any disease you care to name can be attacked in using a genomic medicine approach. And not only that, these medicines can be designed and made in a time on the order of a few weeks, a month or two. Which, when you compare that to 15 years and $1 billion for small molecule, conventional therapeutics, it just changes the whole ballgame.
On a slightly different note, I wonder if you could, on the basis of your substantial experience, talk a little bit about what universities can do from a structural or support perspective to make it easier to develop and sustain relationships between university researchers and external research partners. Do you have any either positive wisdom or cautionary tales based on your experience?
Well, I just think that universities should be taking this more seriously. That there is this societal function. They're supported by tax dollars. The notion that if you have a discovery that's made in a university, that gets out into there's practical use made of it. It should be a priority within universities. Now, there's a certain tension that comes with that, the academic versus industry or business side of things. But it is something that should be facilitated more, and part of that is in the IP side, intellectual property, the ease in which we get technology out of a university and into a new startup. This is still a very cumbersome process for most universities, and I would love to see that smoothed out.
Yeah, I concur with that. I think reducing the friction in that interaction between the entrepreneur company and the university, and I think the goal of the university, rather than trying to maximize every deal, would be to get the technology out there, and the fruits of that and the impact locally to the economy, with the jobs being created, with the royalties coming back to the university, is immense. And having just more technology get out there into the entrepreneurial system should be the number one primary goal.
In addition to getting technology out into the world, there's the issue of getting ideas out into the world through publishing. And another potential area for friction would be around the different timing involved in publishing versus patenting. And I wonder whether you faced any tensions in this regard.
There's ways to avoid those tensions. I mean, once you file, you have a good idea, and it takes often, we'll say 6 months to a year before you get a paper into its final form that you're ready to submit it. But often you have the rough outlines of the data at the beginning of that process, obviously. If you file a provisional patent and then build on that for a patent that will become public within a year, you can just be about writing your paper at the same time. And so I haven't really found that the patenting process has delayed publication in any significant way.
Also on the idea of publishing, what about open source publication of research results? Have there been any difficulties or misunderstandings about the value of proprietary protection of certain discoveries versus publishing in open source forums?
I personally am a proponent to not having the research being done paid by public institutions under name your academic journal firewall. Right now, being an industry and not being part of a university, it's immensely frustrating not to be able to access peer-reviewed journals, basically, full stop. So I don't know how much more to say to that, but I'm definitely a proponent of open source publishing and getting it done faster as well. And I don't think we need to go down the rabbit hole of peer review, but I'm sure there's lots to be considered there.
This question, I think, may be a little bit more for you, Peter, about the decision whether to stay at a university or work full-time in one of your many startup companies. You've obviously decided to remain primarily in the university while keeping your hand in these various companies. So was the ability to pursue high bar research, working closely with partners like James, a factor in your decision to at least keep one foot in the academic realm as you've done this type of research?
Yeah, it's an I mean, I have gone fairly whole hog into industry at one point. I mean, in 1992 and 2004, I was the chief scientific officer of one of the startups that we started called Inex Pharmaceuticals. But I found that it was useful to keep my lab going for one fundamental reason, and it has to do with the teamwork aspect that I was emphasizing a little while ago. If you're in a company environment, you have to be doing work that is really germane and seen as being germane to that enterprise. And I always found that so that's great, but I got to occasionally get interested in things that maybe weren't fully aligned with the company. And so then I found I could do that kind of work in my university lab. And so it was a bit of a release for me to be able to keep that going. And I found it to be, in the end, to be quite useful.
Mm. And James, you described the idea that your company actually worked in Peter's lab for a while. Do you think that co-location was important to maintaining the deep partnership you've described? And did the university make this easy to do? And would you recommend this kind of approach of co-location as opposed to working in an independent location outside of the university campus?
Yeah, I think for a few reasons when you're this early, at that early stage. So we did a lot of the fundamental technology development at that early stage. You're also, as a company, extremely vulnerable. You don't have revenue sources. You're not necessarily attractive for investors. And then we built a lot of our early financing through grants that had a translational component to it, some fundamental components to it. So then I think we were very successful with that. And then we're able to de-risk the technology and the platform until we were ready to go out into the wild.
Yeah, we had some pretty Rube Goldberg sort of instrumentation there as we got to the final nano assembler. I hope you still got some pictures of that, James, because they were.
So much of what you've described, apart from the developing technologies that seem like you were struggling a little bit to figure out how things work, much of this has been a success story. And yet I wonder also whether there were certain things that you might do differently if you were beginning some of this research together, and whether you encountered any barriers that surprised you and that you somehow managed to overcome or didn't manage to overcome.
Starting a company, being an entrepreneur and building it to success is extremely difficult, and there's immense challenges every day. So you're going through a collection of failures continuously at all times, right? And it's a bit of a gauntlet, and people don't necessarily see the inside of the sausage factories. But I would say I think you just need to keep persevering and keep pushing forward and getting to success on the other side. But yeah, there's innumerable failures, challenges that happen, continuously as you're building these things.
Yeah, and I'll just give you one example of how that can occur. One of the companies, Inex Pharmaceuticals, we developed a anti-cancer drug, which we took to the FDA. And at that point, we had a company of 200 people. And, we had every reason to believe this drug would be approved for limited use. But it was just after the whole Vioxx, this was in the mid-2000s, and the whole Vioxx situation had arisen, with the cardiac toxicity associated with that drug that had been approved by the FDA. Anyway, the FDA was very cautious, and they asked us to go back and do some more research, which we did, or more clinical trials, which we didn't have the money for. And so this resulted in a company of 200 people going to 50 people the next day. And that was extraordinarily unpleasant. And so in the end, it turned out that we did work it out in a positive manner, and as much as that we then really switched all our efforts to pushing the nucleic acid-based drugs, genomic drugs, which has been really successful. Yeah, there have certainly been some bumps in the road that are not much fun.
I wonder if any of these bumps were specifically related to funding agencies and whether there's anything that funding agencies could do to encourage the type of collaborative work that you're doing.
I think there's more and more support now, for at the early stage. I'm not sure they could have supported the phase 2, what Peter described there. I think that's a different piece, but certainly funding agencies supporting early stage companies, early stage development, that combination of both basic as well as translational research is obviously critical. But absolutely. And I think now also what we saw with the pandemic is that public-private partnerships are integral to solving deep challenges in society. And the COVID vaccines were developed in part between these deep relationships. So I think that'll continue, and is an important facet of pushing these deep technologies along.
Yeah, I'll just echo that there is now very much more and more awareness that innovation is vital to your economy, right? There are certainly more incentives now that are available than, say, were there 20 years ago. It's still not easy. The more support that's available, the more players you get. So the demand always exceeds the capacity. But on the other hand, if you look, say, the Vancouver area right now, it really is a very vibrant biotech scene, and that 10 years ago that was not the case. So the incentives and the universities are behind you in particular. That can really lead to some good outcomes.
Another vital aspect of this work for society is the training of new generations of researchers and entrepreneurs. And I wonder if you could say a word about your own experience in bringing students, probably primarily graduate students and maybe postdocs, into your labs and into your company to contribute. Can you think of anything that would enable students to participate in such work more effectively?
As an initial statement, people often think that if you're going to do a startup, they really need to get somebody in there who has real business experience, et cetera, to push the enterprise forward. I don't think that's true. I think that taking somebody, say a postdoc in an academic lab who really has ambitions is usually the best way to go. That they can learn they can hire the business expertise. But it's harder to get somebody who is wholeheartedly devoted to making sure that the technology works and has some basic understanding of it. So in my experience, bringing people in at the postdoc level in particular is really a good way to ensure success. If you hire initially a business person, that's never really worked for me. But
yeah, I can imagine. And I would just add, universities play a huge role in providing the workforce for a company like ours, particularly these deep technologies. And so, we hire a tremendous amount of new grads and bring them in, and, further training programs for us in biomanufacturing and other would be very important. But I think overall, the universities and society in general needs to figure out how do we train more and more people. Such a small sliver of society has a PhD. How do we train more and more people, 100x, 1,000 times fold more people to solve the grand challenges of today are going to be solved with these deep technologies. And so we need to figure that out. And I don't think, not necessarily that the current paradigms are going to have this capacity to deal with that. So I think we need to really think about this.
Do you have any suggestions for innovative paradigms that might facilitate the involvement of students?
I don't know. Like I'm not. Yeah, I don't think I'm an expert in education in that way. But how can we make 100 times more PhDs? Right? How do we accelerate these? Be it through virtual learning, whatever it is, the different ways that we can do it and get in a more scalable educational approach would be, I think, very beneficial for society.
Speaking of virtual, I was just going to follow up on your point, James, about virtual learning. One of the things we've learned during the pandemic is that we can do a lot of our collaboration from distant locations. So it may be that you don't have as many PhDs as you need in Vancouver, British Columbia, but maybe there are PhDs elsewhere in the world who could be participating. What do you think about the possibility of working with partners who aren't geographically close in collaborations like yours? You both happen to be in the same geographical location, essentially, but could much of what you do actually occur virtually?
We have many employees that are virtual. I would say in terms of training, we also have set up our own NanoMed U online training program for people to understand how to make these types of drugs better. Some work definitely needs to be co-located. You know, science on the bench type work needs to be in a place where there's a bench. So either you can create, you know, different pockets around the globe or have them come to your locale. Absolutely, but I think again, you know, more and more people developing these advanced technologies will create more startups, will create more jobs. It's a virtuous cycle, I think, and you know, one we cannot underinvest in.
Yeah, thank you.
Yeah, and I mean, just to follow up on that, Scott, the whole notion of people working virtually, I totally agree. That really has changed the ballgame. Working with people that are, where are we? I mean, you're probably a couple of thousand miles away from me, and you know, we're all used to that now of having conference calls with people in Europe or Asia or wherever it might be, and that so they can be really quite productive. So I think COVID has really, you know, this era has really changed the ballgame in terms of expanding the possibilities of taking advantage of expertise, no matter where it is.
Yeah. Well, we've certainly taken advantage of your expertise today and greatly appreciate all of your thoughts and insights. I wonder if you could just take a couple minutes now and offer any final takeaways.
As James will attest, I mean, as we've indicated, there are certainly moments when you try this, you know, translational kind of research, trying to get things into the commercial world and build companies, et cetera. There's certainly moments where you think, boy, oh boy, why have I done this? You know, there's lots of things can give you hesitation. But on the other hand, it's hugely enjoyable, both from a team point of view and then as being part of a team that actually achieves something, whether it be, you know, enabling a new medicine or a new instrument or whatever it is. And so that is, it's a wonderful journey to go on.
Excellent. Yeah, I totally agree. The other thing I think I'd like to impress upon everyone is that medicine is at a transformation. The biggest revolution of our generation is happening right now, and it's a unique opportunity of productivity that will happen in society to benefit patients, and a unique opportunity for folks to get involved with these technologies. It's akin to you know, when physics enabled computers to go from analog to digital. It's the same revolutions happening now in medicine, which is just fantastic.
Thank you so much, Peter and James. And thanks to everyone who's attended today's webinar. Please stay tuned for announcements of future events in this series.